Exhaled Aerosols by Faizan.
Okay.
Thank you.
Hello. So the title of my PhD is The Structure and the Composition of Exhaled Aerosols. Is to give you a little background, as you all know very well, especially after COVID, that airborne transmission is a major route of disease spreads of respiratory diseases in humans. Now these are emitted through exhaled aerosols and droplets, which we emit during various respiratory maneuvers, from like talking to speaking to coughing and sneezing. And they have a we emit them in a wide size range from as small as 0.1 micrometer to as big as more than 100 micron. But the majority of them are from 0.3 to like 10 microns. And the concentration is very varied in a person, is like 1 order of magnitude, and it's varied in between person by order of like 3 or 4 order of magnitude different. And the thing is that when we emit these droplets into the ambient environment, so they can either be directly inhaled by other individual, or they can be deposited onto surfaces, which then indirectly by emitted by inhaled by other individual or got contact with them. So these droplets and aerosols are very complex particles, mainly containing water, salts, lipids, and proteins. And when they are emitted into the ambient environment, so the conditions in the ambient environment are different, so they go rapid evaporation, which result in the crystal formation in these droplets. And now the structure and crystal formation in these droplets is very important, but the detail on them is very limited. There are remarkably low amount of images of them, and the characterization is very much limited. So that's why we wanted to look at them, because they are very important for the disease prevention. So which brings us to the objective of our project. What we want to do is to develop imaging and characterization technique to look at exhaled aerosols, and then use the same techniques to look at synthetic respiratory fluids, which are mainly used in these kind of studies. But we are not very sure. There's still a debate in the literature whether they are representative of exhaled aerosols or not. And then to try understand the effect of the ambient environment on the pathogens that are present inside these droplets, and of the local environment within a droplet, to see how that affect the pathogens inside them. So what we will do, we use different microscopy techniques ranging from optical microscopy to more specialized techniques to look at these droplets. But for today's studies, we will be only focusing on the work that we have done with these two techniques, to mainly look at the influence of composition on the crystallization of simplified synthetic lung fluids, which mainly contain sodium chloride, mucin, and DPPC as salts, proteins, and surfactants, to be like the simplest form of a synthetic respiratory fluid. We would look at the influence on the crystallization morphology, the nucleation of sodium chloride, and a technique that we developed to capture exhaled aerosols. So what we do in our system is that we have developed one where we put our droplets on a cold stage under a microscope in a temperature and humidity controlled chamber, to look at the crystallization dynamics of the droplets. So yeah, so this side is mainly the RH control system, where we put in dry and humidified, the ratio of dried and humidified nitrogen to the system to control the RH. This is the chamber under the microscope, and we have put in a hygrometer at the output, just to be sure that the RHs that we are putting into the system are actually correct or not. This is what the chamber under the microscope looks. So you put the droplet on the cold stage, which goes airtight with the objective of the microscope, and then you can control the RH into the system. This is how we have been atomizing our droplet using a nebulizer into a chamber, and then we put a glass slides in the neck of the chamber to get a distribution on the slide, and then just transfer the slide into the chamber under the microscope. So this is what usually you would look under the microscope when you run the experiments. Actually, this is what you would look in the camera on the screen. We record videos from it. In the microscope, you get a more wider and view of the particles. And what we can do with the system is that we can either increase the humidity, or we can decrease the humidity, relative humidity in the system. So when you increase the relative humidity on the particles, so at a certain point they would all become liquid, which is called as deliquescence. Now, deliquescence of these particles is rigorously reported in the literature, and it's a specific RH for sodium chloride. So it's sort of a calibration for a system that our system is working well, and the readings that we are getting in our systems are in very much in agreement with the literature. In the images, as shown on the previous slide, you could clearly see when you run through these ramping experiments, the morphology of the droplets change, and you can clearly see when they nucleate and become crystals. The other thing that you could do with these experiments is that then you can decrease the RH when the droplets would eventually crystallize. This is called as the efflorescence of the crystallization relative humidity. So we did that for sodium chloride, and as you can see that sodium chloride um, crystallizes within a range of relative humidity from like around 50 to 44% RHs. And when we add in mucin into the solution, so the crystallization relative humidity of sodium chloride is delayed a little bit, but there is no significant impact of the different concentration of mucin that we used with one specific concentration of sodium chloride. If you look closely, there is still a trend as you increase the concentration of mucin a little bit, it's delayed a little bit more, but generally it's on the same thing. Then we add DPPC to the solution. So we kept the concentration of sodium chloride and DPPC constant, and we did the same experiment with 3 different concentration of mucin, and we saw that since we are adding a very little amount of DPPC into the solution, there is no significant impact on the crystallization rate of the droplets that we have. So when we look at the rate, we thought that there might be, because with the optical microscopy, you cannot see very good images. So we wanted to see whether there is any impact on the crystal habit of the droplets. So this is usually what you have seen as sodium chloride crystals. But when you add mucin to the solution, so you get some of these dendritic structures with this is a droplet with the lowest concentration of mucin. So you could see like thicker dendritic structures with branches which are not very definite, but as you increase the concentration of mucin, the crystals become thin and the branches become more finite in them. Just to give you a little takeaways from the work that we have done in here, so the mucin is impeding the crystallization rate of sodium chloride and is changing the crystal behavior of sodium chloride, while DPPC is not having any significant impact on the rates. When we were doing these experiments, so we were looking at data for sodium chloride, and we found out that in literature, there are literally no experimental nucleation rates calculated for sodium chloride. So we thought that why not do it? So in our setup, we could do that by 2 ways. One is like we can keep the relative humidity constant and let the droplet dry, run the experiment for a very longer time. And the other thing is to ramp the experiment on a specific rate periodically and look at the droplet that crystallize. So under the isohumid conditions, if you put the natural log of the decaying rate of the droplets to the total number of the droplets against the volume and time term, so you get a straight line on the graph, and the gradient of that graph would give you the nucleation rate of the droplets. In the ramping experiments, so we are changing the relative humidity, so the nucleation rate is much faster. So what we did was we divided our droplets into different bin sizes to be for the, as we are using an average volume term. So that's why we divided into different bin sizes to look at the nucleation rate. And as you could see, the nucleation rate from the isohumid experiment and ramping experiments agree very well with one another. And as expected, as the bigger droplets would nucleate on higher RHs and smaller droplets would nucleate on lower RHs. The 2 values that I have given as a reference from the literature are the values for nucleation of sodium chloride, but they are not experimentally calculated values. They are more like simulation values. Now, if you remember, I told you that we would also the core to the project is capturing exhale aerosols from human beings. So we have developed a system for capturing them where we would be using a modified snorkel mask, which would be done by an individual and would do these respiratory maneuvers into the mask, and from where the the particles would go into a steel drum, and we would collect from the steel drum particles on a cascade impactor, which is basically a six-stage size classifier, originally designed to collect bioaerosols from the ambient environment, where you put your agar or culture media in a petri dish, and then you leave the system in the ambient environment to collect aerosols on it. But we would be using it on the other way around, and we are not using agar plates on it, but we could use whatever media you want. For example, we have been using SEM stubs in with these experiments. You can use polycarbonate filters and TEM stubs as well in this system. So we ran some test runs with this equipment with sodium chloride to see whether our system is working or not. So we could see good distribution of particles. These particles are directly collected on SEM stubs, and we could see that we could get a good distribution on different stages of the system. It seems like the system is working, so that's good. So in summary, so we worked on the sodium chloride, mucin, and DPPC simple synthetic lung fluid system to look at the nucleation and crystallization in these systems. We talk about the nucleation rate of sodium chloride and the equipment that we used for the collection of exhaled aerosols. In the future, we are planning to use this equipment to collect exhaled aerosols from human beings. We have put in an ethical review for it, which is under process. Hopefully, it will be granted to us, and we would be using the same equipment to then atomize synthetic lung fluids and apply the same characterization technique to them to form a comparison between the real droplets and the synthetic droplets. And there's also potential to use single droplet levitation techniques to put in surrogate viruses in the droplets and look at the drying dynamics to see how that is impacting the viruses or the pathogens present inside them, and where are they ending up in the droplets. And thank you very much. And I would like to acknowledge my group and all the people who helped me develop these rigs and the system. Thank you.
Right, any questions?
So it's a quick talk. I found it fascinating, the results, especially the RH steps you took. I was just wondering, was the RH stable across, and how was the uncertainty? Was there an uncertainty range for the RH sensor, the relative humidity sensor you used?
Yeah, so this was the main difficult thing with our system. We have worked a lot on it. So because you need to keep the temperature very constant for the RHs, it's very much sensitive to the temperature. So the chiller that we are using are very good, and we control the temperature on the cold state as well. So there's a chiller connected to it, and there's a heater connected to it. So it doesn't fluctuate that much. There is only fluctuation of 0.1%, which is still, and we are changing the RH with changing the flow rates of the dried and humidified airs. So the temperature I kept very constant, and the initial result that we were not getting the results that we wanted, and we figured out that our RH is not very constant. So we have thermocouples, and we have the hygrometer at the output, which give us a right on the time display of the RHs if it changes.
Hi, that's a great talk. I was just wondering whether you're nebulizing into this chamber and collecting the particles on the glass slide. Are the particles drying in the air, or are they landing on the slide and then drying on the slide?
So
is that a true representation of the aerosol? Yeah.
It depends on the RH in the lab and the temperature in the normal days. So when you get the droplets on the slide and we transfer it to the system, so then it doesn't matter because we can then we can then change the RH. So if you want them droplets, we can increase the RH, and if you want them crystals, we can decrease the RH.
Is there any other techniques that you could use to recreate that?
With this system, so we need to put the glass slide into the system. So it needed to be put on the slide and then transferred to the system. So like developing a technique where you could directly, put in a nebulizer into this chamber where you could directly put the droplets on the slide in the system. So then you would avoid all these things. But it seems very sophisticated, and the system is working very well. So we have been using this.
Yeah, it's really good images.
Thank you.
Fine, a quick one. Faizan, have you looked at what structure you get if you just try the mucin droplets on their own? Because I'm curious whether that dendritic structure is seen in just pure mucin droplets.
We haven't done SEM for that, but from the optical microscopes, the structures are not dendritics. It's sort of like in the optical the resolution is not very good, so I'm not able to explain it very well. But we have plans to look at the SEM images for mucin to do that. But from the optical microscope, I can tell you that they don't look like the images that we have for the mucin and sodium chloride together.
Great, I think maybe we move on to the next speaker now. Thank you.